Good turnout. Very nice to see. Okay. Well, welcome everybody to College of Science virtual seminar. I'm Michael Kaufman. I'm the Dean of the College of Science. And it's a great day to be getting together late on a Friday afternoon for a nice interdisciplinary talk from Dr. Wendy Lee. Let me tell you a little bit about Wendy. Wendy is an assistant professor in her third year of that appointment at San Jose State University. She's also the graduate advisor for the Masters in Bioinformatics program in the Department of Computer Science. Wendy earned her PhD in bioinformatics from UC Santa Cruz, just over the hill, a master's in molecular biology and microbiology right here from San Jose State University, and a bachelor's of math and computer science from the University of Waterloo in Canada. Dr. Lee has over a decade of industry experience developing bioinformatics analytical pipelines and enterprise software. Her research interests include developing and applying bioinformatic methods to analyzing next-generation sequencing data for genomic, transcriptomic, and metagenomic studies. She also has a passion for fostering the success of women and girls in STEM fields through training, networking, and mentorship. She's currently the faculty advisor for the SJSU Girls Who Code College Loop. Wendy's going to be telling us today about her work on the following: the impact of BPA, which is one of those nice plastic components that we all know about, the impact of BPA exposure on neurodevelopment at the molecular level. Just a couple of ground rules for our talk. I will be monitoring the chat to see if anybody has a question during the talk. If it looks like something that I should interrupt our speaker for, I will do so. If it looks like something that can wait till the end of the talk, we'll make sure that we leave time for people to ask questions at the end. And I'll, at the end of the talk, if you're asking a question, I'll allow you to ask the question yourself. So without further ado, Wendy, take it away.
Thank you so much, Michael, for a great introduction about me. And I am so happy to be here to tell you about one of my research projects that I think is very relevant to all of us. Like Michael said, BPA is this chemical that is in almost everything that we use in plastic bottle, plastic containers, tin can linings, and all that. And how does that, such ubiquitous, type of chemical that we can't really avoid, and how does that impact us on a neurodevelopmental level? So let me move forward. So just to put things in perspective, like why are we talking about neurodevelopment, is because more than 1 in 10 children in the United States are diagnosed with a neurodevelopmental disease. We also call it NDD because it's shorter. So NDD is known to be caused by combination of genetics and environmental factors that leads to changes in the brain development while the fetus is in the womb. So what happens in the womb sometimes actually lasts a lifetime. And because we're talking about environmental chemicals, there are just so many, but the ones that we expose to often that we can find in plastic bottles and aluminum cans is something called the bisphenol A, BPA. I'm sure many of you are now using BPA-free bottles and thinking that, oh, I'm okay, I'm using BPA-free stuff. I don't really have to worry too much. So just wait until the end of the talk, then you might think otherwise. So without giving you too much, the actual story yet, just want to kind of, again, put some perspective on how much BPA are we looking at yearly worldwide. We produce about 8 million tons of BPA, and makes us the largest volume of chemicals produced. And it is a reproductive, developmental, and systemic toxin. And so we need to, really know how this chemical works, right? So on the right side is showing you the chemical structure of BPA. It's structurally really similar to a female hormone estradiol, and it can easily cross placental and fetal blood-brain barriers. So how does BPA actually impact our body then? If it structural looks like a female hormone, it's actually known as an endocrine disrupting chemical, EDC. And if you want to know what is endocrine system anyway, endocrine system made out of all the body's different hormones. Hormones regulates all biological processes, including development of the brain, nervous system, growth and function of reproductive system, metabolism, blood sugar levels, you name it. So if it impacts our whole systemic level of regulation, you can imagine that absorption of BPA actually has been found to lead to prostate cancer, breast cancer, obesity, diabetes, glucose intolerance, insulin resistance, and infertility, a bunch of things. So that's why there's BPA free bottle, because people know that BPA is not very good. Let's focus on brain then, because we talk about, when we started, we talk about, oh, how many children actually just diagnosed with neurodevelopmental disease. So let's look at how our brain works. We want to focus on two parts that it is known to be affected by BPA exposure. One is hypothalamus. Hypothalamus is somewhere in the midbrain. The purple dot that is pointing is hypothalamus. It controls emotion, regulates body temperature, and control crucial urges such as eating, sleeping. And hippocampus is kind of underneath. The red part is also involving in memory and learning. So in animal model, research has shown that prenatal exposure of BPA actually decreased hippocampal myelination, which is like the insulation of the hippocampal area, which leads to impaired cognitive function. And it also disrupts gene expression in hypothalamus. Even at a low doses of BPA exposure, it leads to behavioral changes. And the disruption of sleep patterns and social interaction and neuroactivities are observed in pregnant animals while carrying the babies when the mom is exposed to BPA. But this kind of sleep disruption and social behavioral changes actually is passed on to the children, and multiple generations after we still see this kind of phenotype that is displayed in the pregnant animal. So the impact of BPA is pretty widespread in neurodevelopment, behavior, and also neurodegeneration. So not only in neurodevelopment is being, impacted, such as called, known to associate with autism spectrum disorder, attention deficit hyperactivity disorder. It also had known to have consequences in neurodegenerative diseases such as Parkinson's, amyotrophic lateral scoliosis, and ALS and multiple sclerosis, MS. So today we're going to focus more on the developmental side of things. So we want to look at how about autism spectrum disorder, because a lot of children has been actually increasing in number of diagnoses. I'll show you in a little bit, but let's look at what is autism spectrum disorder. It's a spectrum because there are, high functioning autism level, which is they need support, but with support from, professional people who can help the patient improve on their communication skills and maybe reducing the repetitive behaviors. But as the spectrum goes to the right side, more severe autism patients actually requires very substantial support. And even with that, their communication skills is not improving, and also their repetitive behavior is so severe that impairs their daily activities. So if we look at the prevalence of autism spectrum disorder, just from 2000 to 2016, it keeps climbing. This bar chart is showing prevalence of autism spectrum disorder per 1,000 children of 8 years old children and older. And between 2000 and, between 2000 2016. And, it just really, it's a upward slope. And you're thinking, well, what about the burden, financial burden and social, social economic burden? In 2015, it cost US $268 billion to care for children with autism spectrum disorder. And it's projected to be 461 billion in 2025, which is a lot. So there's this gene by environment hypothesis. We know that autism spectrum disorder is a complex disease that both, are impacted by genetics and also environmental factors. And there are over 100 risks genes that have been identified that is associated with autism spectrum disorder, but the identification of the environmental factor is a little bit behind. Environmental factor includes industrial chemicals, drugs, maternal hormones, etc. So that leads to the motivation of this research project, because the number of neurodevelopmental disorder diagnosis just increase year after year, especially for autism spectrum disorder. And with the prevalence of BPA in, common materials, and seeing that there's, evidence supporting that BPA actually plays a role in influencing neurodevelopmental process, we want to spend more time to take a look at, how does that actually impact our brain during development at multiple levels. So this is the team that I have been working with. It's a great team. And I don't have enough space to show everybody, so I'm only showing the key team players. This is a team that we investigate the neurodevelopment impact of BPA exposure in Drosophila in wild type flies. Drosophila is a common fruit fly that is used as model organism, if you don't know yet. I'll tell you why we use it in a bit. But we also look at fruit flies that actually have a genetic background, which is Fragile X syndrome disorder. And so we want to see how BPA also impact flies that already have a mental disorder. So we, in our team, we have a group of scientists looking at behavioral and cellular level impact of BPA exposure, led by Dr. Kimberly Milligan from CSU Sacramento. She is an associate professor in biology. And her student, Yuen Nguyen and Brendan Tinsley, and a few other students have been working really hard, observing flies and growing them. And yeah, it's a lot of work, and they did a really awesome job. And recently, 2021, this year, early January, they published a paper on this part for behavioral and cellular level observation of BPA exposure impact on neurodevelopment. And of course, we don't want to just know 2 levels. We want to know deeper level, the molecular level. So we have in San Jose State with the bioinformatics master's program. We have some really awesome students that have been working on this project. Eden Johnson, who just graduated a couple months ago from the master's bioinformatics program. And she was analyzing the RNA sequencing data coming out from this study. And Rashid Ragulan just joined my lab, and she'll be studying the RNA gene expression with the Fragile X syndrome genetic background. So this is the paper that just got published early this year, super exciting. And so now let me tell you about the experimental design of this whole project. So we're using Drosophila melanogaster, which is the common fruit flies. We have 2 different types. We have the wild type, W1118, which is, just the name, and a mutant strain that has a Drosophila FMR1 mutation. I'll tell you a bit about FMR1, what it is, but just keep that in mind right now. So it's a mutant that has a Fragile X syndrome. We expose these flies, both wild type and mutant, with different concentration of BPA or dosage of BPA versus no BPA exposure. And then we observe the behavioral phenotype, including larval locomotion, grooming, courtship. the cellular level, we want to look at how the neurodevelopment actually impacted by BPA, including axon guidance and also neural stem cell development. Third, we look at the molecular level, which is the gene level and the pathway level using RNA sequencing. And we look at the differential gene expression between the two groups, wild type versus BPA without BPA, or mutant with BPA or without BPA. So to see the impact of BPA, right? So let's look at what is Fragile X syndrome. Fragile X syndrome is a single gene disorder, meaning it's this one gene causing this mental disorder. And in human, Fragile X syndrome is actually occur in a gene called FMR1, named Fragile X mental retardation 1. And this gene in patients with Fragile X syndrome is actually turned off. And I'll tell you in a bit why it is turned off. So, and interestingly, about quarter to half of the people with Fragile X syndrome are also diagnosed with autism spectrum disorder. And that is why we use this Fragile X syndrome to understand how BPA influence autism spectrum disorder genetic background as well. So Fragile X syndrome has this gene, normally FMR1 encodes, like people without Fragile X syndrome, FMR1 encodes FMRP, which is the protein that regulates neural stem cell proliferation, axon guidance and outgrowth, synaptogenesis and synaptic plasticity. So everything about neurodevelopment, this protein actually regulates and kind of bind to other parts of the RNA to control different genes to actually help normal neurodevelopment. And how does that mutant FMR1 turn off? That happens when in the upstream of FMR1 gene, there is normally 5 to 50 CGG repeats. So it's a tandem series of CGG, CGG, CGG. In the mutant version, so with the Fragile X syndrome patients, this repeat actually expanded from 200 to maybe over 1,000. So when there are so many CGGs, actually it will cause hypermethylation, which is a epigenetic regulation type of thing, that tightening up the DNA so that the RNA polymerase cannot come in to make transcript. So basically the gene is shut close, cannot be expressed. So no transcript, no messenger RNA, no proteins. And so the regulation of all these neurodevelopment is all interrupted. So now let's talk about why we use Drosophila as a model. First, Drosophila actually has the same FMR1 gene that is having the same function as the human one, which is one of the reasons why we have the Fragile X syndrome Drosophila model. Second thing is, in general, fruit flies is a great model organism because it's low cost. It has a short life cycle. It's easy to maintain. They're small. They're not like big organisms that you can feed a lot of food. And it's easy to expose them to different environmental chemicals to do toxicology tests in the lab. And most importantly, 61% of the disease-related genes in human, there is a gene that is in the fly that actually do the same thing. So we call those orthologs. And so it's easy to actually study human genes in an animal model, just like the NDD-associated genes FMR1. So now let's look at some of the results. We'll focus on the behavioral level impact of BPA, and then looking at the cellular level, and then look at the molecular level. So let's go with the behavioral level first. We look at the larval locomotion. So this larvae is actually, before they turn into adult flies, they're looking like little, larvae, and they move up and down. They can turn side to side. So in order to see if they have higher activities or hyperactivities, we measure the number of times they do this peristaltic contractions, and also the number of times that they change orientation of their body. And it turns out that with BPA exposure, like, so this plot is actually showing the black one is controlled with no BPA. And we have two groups, the wild type and the mutant type, the Fragile X syndrome one. And then the blue and the orange are at different doses of BPA exposure. So with BPA exposure, we see that there's increase in peristaltic contraction for the wild type, but not much changes in the Fragile X mutant. That is the up and down motion. But for orientation change, there's also substantial increase in the wild type in orientation change after BPA exposure, and a bit of the decrease in orientation change when the mutant is exposed to 0.1 millimolar BPA. So it's more like a partial decrease, because for the peristaltic contraction, there's not much changes, and for orientation changes, there's some substantial decrease from control versus 0.1 millimolar. So that's one observations that we made. And then another thing that we are looking at, does BPA actually change the amount of time that they spend grooming themselves? So with BPA exposure, the blue and the orange bars, the gray bar again is controlled without BPA. So wild type flies after BPA exposure, there's increase in grooming time. And then for the Fragile X background, there's a decrease in grooming time. So that's another observation. So it's different. And the third behavior we're looking at is courtship. So courtship is measured using something called a courtship index. It's calculated by the amount of time that the flies, or the female and male flies, when they're put together, that the time they spend doing this all these 6 different activities, anytime they do any of these is counted, and then divided by the total amount of time is they're being observed. So that will give you the courtship index. So when we look at the wild type flies, when they are exposed to BPA, they have decreased in courtship index. While the mutant, they started off with pretty low courtship index, but then the exposure of BPA doesn't really change anything. So now we know the behavior are different with BPA exposure. How about at the cellular level? Does it change how the brain develops? So at this stage, we're looking at the larvae and see if whether neurodevelopment is actually impacted by BPA exposure. One way to look at whether the brain is affected, the development is affected, is by looking at, is there any abnormal axon guidance, phenotype. So abnormal axon guidance is actually a typical cellular phenotype associated with a number of neurodevelopmental diseases. And in Drosophila, axon guidance can be examined by looking at the adult's mushroom body, which is like the midbrain of the fly. So it's circle, it's kind of box right here. So the mushroom body is actually a bilateral symmetrical structure illustrated inside the box. And it is has this 3 different lobes. It has the alpha, beta, and the gamma lobe. When there is a axon guidance disruption, usually we can observe that by looking at the midline between the 2 beta lobes. This is normal. The beta lobe is not crossing the midline. But if there's axon guidance disruption, then you see that the beta lobe starting to kind of cross over the midline right here. And they're moderate, mild, and then moderate level, the midline is even more pushed by the beta lobe, and then the beta lobe looks almost like it's fused together. And then in severe case that, it's completely kind of run into each other, the beta lobe. So this is how we measure if the axon guidance is disrupted by the frequency of the midline crossing. So in this case, we noticed that there in the wild type with BPA exposure, 0.1 millimolar and 1 millimolar has increased the frequency of midline crossing, meaning there's increased the amount of frequency observed of axon guidance abnormality. But then when it goes up to like 2 millimolar, it seems to have dialed down a little bit. It's not as much. So, but for the mutant Fragile X background, we see that when they're exposed to BPA, they actually have less midline crossing. So the black bar is the severe cases. So, it seemed to get less and less. So it seemed like BPA might have a protective mechanism for the Fragile X background from getting abnormal axon guidance. Another thing that we looked at is looking at neural stem cell development, whether it affected by BPA exposure. So we look at in this, the larval brain, we stain it with anti-DEPN to mark the neuroblast and the mature intermediate neural progenitor cells. So all these developing neural stem cells are all marked as green dots. And by looking at the wild type after BPA exposure, it didn't seem to have decreased the number of neurodeveloping cells and neural stem cells, while the genetic background has substantial decrease in the number of brain lobes and also neuroblast stem cells. So now with all these cellular level and behavioral level, we don't exactly know what are the genes that are impacted, influencing or causing the phenotype. So in order to go to the molecular level, the gene level, we have to extract the RNA and send it out for sequencing to get a whole genome-wide view of what are the genes that are actually involved or being impacted. So same thing, we have wild type and mutant flies. We expose them with or without BPA, and then extract the RNA. So the RNA extraction part is done by CSU Sacramento, Brandon Tinsley, and we send out the RNA to UC Davis Genome Core and also GeneWiz, which is a commercial company, to do the Illumina high-seq sequencing. And with all the sequencing data coming out, we perform all the analysis on the high-performance computing clusters in San Jose State and the College of Science. And so now with the data, we do two types of analysis. First, we look at what are the genes that are differentially expressed between the two groups, BPA exposed and no BPA. So that's called differential gene expression analysis. And with all the genes, so many genes, can we find out, are these genes working together in a pathway, a molecular pathway that actually related to some kind of biological processes or cellular processes and all that. So that's called gene set enrichment analysis. And the wild type gene expression analysis and gene set enrichment analysis is done by Eden Johnson. And Rashi is the one who's going to do the mutant. So this is a cliffhanger because we do not yet have the mutant RNA-seq results yet, but even just the wild type results is amazing. So hopefully you will agree. When we first got the data, we have millions of sequencing reads and we have to align it to the reference genome and a lot of data analysis has to be done. And then out comes this beautiful plot and tell you how many genes are actually differentially expressed between wild type with no BPA versus wild type with 1 millimolar BPA exposure. So we found 1,040 genes that are expressed differently between these two groups. And we measure using a p-adjusted value less than 0.01 to say this is statistically significant. This plot is showing you the log2 fold changes, meaning how different are these expressions. So the fold change and take a log2, so that will be the number, and they're binned into, these are the frequency, how many genes are actually differentially expressed by how many log fold change. And so we're like, okay, majority of them are, maybe log2 fold change. How about the extreme ones? How about the ones that are on the distal ends? What are these extreme changes? What genes actually change so much between the two groups? So we picked the top 10 and we see that, wow, just the top 10 extreme fold change, we see something that we recognized earlier. Myo, which is related to biological processes for mushroom body development. Tau is related to nervous system development. ARU is involved in memory, olfactory behavior. EXN is involved in regulation of neurotransmitter secretion. So while that's great, how about the rest of them? Like we have 1,040 genes. We don't want to lose all these information. So one of the beautiful things is that in bioinformatics, there are a lot of databases, there are a lot of people putting together a lot of information, and we can use them to kind of make connections. So with a bunch of genes that we have found, 1,040 of them that are differentially expressed, is there any of them actually are in a set that is involved in a specific pathways that is doing certain things, such as, is it doing neurodevelopment? Is this set of genes involving in breaking down glucose? So by running this analysis, gene set enrichment analysis, we can identify molecular pathways involved based on the set of genes that we have. So there's a lot to look at, right? Whoa, what are all these gene ontology things? So these are all the pathways involved in the cellular component, that all the genes that we have, these are genes that are in the 1,040 genes, and of those, a subset of them are actually belonging to responsible in the pathway doing, regulating or work in a synapse. And then synapse is part of the neurotransmitting, gap. I'll show you in the picture in a bit. But you see synapse, presynapse, postsynapse, there are a lot of synapse, and the dendrite terminus, all these underlined ones have direct known association with neurological processes. And this is pathways that we identify using the differentially expressed genes, comparing with or without BPA in wild type larvae in the brain. So you're like, well, I don't know what are synapse, presynapse, what are those? So just bring you to our brain. Let's look at a nerve cell, also known as neuron. So a neuron is a really interesting cell that has a cell body, and then with a lot of like tentacle things called the dendrites, followed by a long thing called the axon, sort of like almost like a wire that has insulation, and then leads to the end is called the axon terminal. So when there's a signal from the brain needs to be transferred to somewhere else, like I want to move my hand, my brain will be transmitting signal from one neuron to another neuron through this axon. And every connection from one neuron to another neuron, there's this junction where the neurotransmitter has to go pass from one side to another neuron, is called the synapse. So this is this presynaptic neuron that have the neurotransmitter going from one neuron to another neuron. And this is the dendrites where it's receiving the tentacle thing that receive the neurotransmitter from one neuron and then pass down to the next one. So that's how the brain actually sends signal through our body. So now let's focus back on, well, we saw these pathways involving in synaptic activities. What exactly are the genes? What are they? And what are the expression level? So this is a very typical heat map. We call it heat map because this is an illustration that shows you how relatively the gene expressions are different between the two groups. So we have the BPA exposed group on the top label, the purple, and then the control group, which is no BPA exposure on the bottom. And so you have a bunch of genes that has relatively higher gene expression level, and these are all the genes. And some of the genes that actually have lower expression level in control, but higher expression in BPA exposed samples. So, and all these are different samples that we have. We have 4 replicates for each group. And for doing this type of analysis, it's always good to have replicates so that we can see if there's a pattern going on. Of course, among the samples, there's some deviations and, it's normal. That's why having the more replicates, the better. And this is the synaptic activities looking at the gene ontology group, the group of genes that is involved in synapse. And then we have the pre-synapse and the post-synapse. Similarly, we're looking at 2 groups and see relatively the gene expression, how different they are. And these are the genes. Remember, we started off with thousands of genes and then ended up with 1,040 that are significantly differentially expressed. And now we're only down to a handful that we can actually do further study in the lab and maybe knock down a gene in the lab and see if there's an impact in the neurodevelopment pathways. So we also looked at the gene set enrichment for biological processes, see if there's any genes that are differentially expressed that, related to, a bigger pathway. So the top one is actually nervous system processes, which is really encouraging because what we're looking at right now is wanted to see if the gene expression differences we're looking at is really impacting neurodevelopment, right? So nervous system processes is definitely related. And these are the genes that are differentially expressed that is involved in the nervous system biological processes. And we're going to further study these genes in the lab. And just to kind of, the gene names are very small in the heat map. You're like, what are these genes doing? Just to pick a handful of them so that you can see those that are upregulated, meaning they are higher, expressed higher after BPA exposure, are the top 3 genes that are involved in sensory perception, sleep regulation, sexual behavior, controls of muscle, neuromuscular junction growth, and a few genes also downregulated, more downregulated genes after exposure is, courtship behavior, mushroom body development, insulation of the nerve cord, and synaptic organization, maturation, social behavior, and all that. So they are all really, really relevant and kind of echoing what we have seen at the cellular level and also the behavioral level. Now we're down to this molecular level and be able to find specifically what genes that could be involved in the changes that we experience observing. So there's still work to do. And this is the cliffhanger that, I cannot show you the molecular, level of how BPA exposure impact the Fragile X mutant larvae yet. So I will come back next time to show you the results. But we also have done a few experiments to expose the flies with other BPA analogs that are also often used to replace BPA, because now we have BPA-free bottle, we need to still use something to make plastic. So in conclusion, through RNA sequencing, we were able to identify genes that provide explicit support and evidence that BPA exposure does influence neurodevelopmental genes expression and pathways. And so we have built a bioinformatics pipeline to analyze RNA sequencing data coming out from this project. And now we're exposing the flies with other BPA analogs and doing other environmental, chemical testing. So BPA analogs, often are known as BPF and BPS, a common substitute for BPA, and they are now often used in BPA-free products. So we don't know yet if they are really harmless or harmful. So I'll come back and tell you next time. So I want to give an acknowledgement to my collaborator, Kimberly Mulligan, who is the PI of the group, and Ewan Nguyen, Brendan Tinsley, helping with getting the biological sample and also extracting the RNA for sequencing. And my lab, Eden Johnson, Rashi Regulin, for helping on this project, and of course other students in my lab. And we went to Santa Cruz for a picnic this summer to celebrate the celebration of Eden. So in the middle is Eden, and Ellen also graduated, and so celebrating the graduation is missing students, but glad that we were able to get together. And of course, funding support. Really fortunate to have CSUPERB Investigator Research Award to support this sequencing effort. So thank you for coming to the talk. And this is my email. If you have any questions, interested in talking more, feel free to email me. This is a needle in a haystack story. Very excited that we were able to find genes that are really kind of correlate with what we have seen in different levels of BPA exposure. Yeah.
Thanks, Wendy. Great talk. It's really spectacular to see the behavior and the genetics linked up so carefully. I'm going to start off with a question about applicability to more complex organisms, right? I mean, one of the amazing things about the Drosophila system is that you can so clearly link those things. How easy do you think it's going to be to make a similar kind of connection in humans, for instance? Like if you examine the genetics of someone who shows autism spectrum disorder, are you going to see similar kinds of genetic markers? Can you link it directly back to the BPA?
Yeah, so I think, this is definitely something we need to look into. Of course, model organism can give us prediction, but at the end of the day, we still need to have actual sequencing data from humans and actual evidence from humans in order to know, are we really looking at exactly the same thing? But because we're looking at genes that are orthologs, meaning they have the same function from two different organisms, that they're supposed to be
Right.
related. So hopefully what we're seeing in the model organism is something that we can actually map back to humans and continue the research that way.
Cool. All right, the first question comes from Frank's PC. Let's see if there he is. Frank, I'm going to allow you to talk if you want to go ahead and turn your mic on, and you can ask your question about RNA. Go ahead. Frank, are you there?
Yeah, sorry.
there we go.
Hi, Frank.
My microphone always has these sort of problems.
That's all right.
So I asked a question in the chat. I said, how was the RNA extracted from the fruit flies?
Good question. That's an excellent question. So what happens is that, in the fruit flies in the brain, you have all kinds of genetic materials, right? With DNA, RNA. And so there are molecular kits that you can purchase from company, and it can isolate all the total RNA, so all the RNAs. And then we're interested in knowing the messenger RNA because we're looking at gene expression, right? So there's also kits that actually have poly select, which is sort of like a little probe, can fish out all the mRNA, and then we can enrich all the messenger RNA and send it out for sequencing. So the reason why the FMR1 genetic background RNA sequencing data come in later is because the larvae is actually really sick. So it takes a long time, especially also during COVID, and it's really hard to have enough sample to extract enough RNA to send out for sequencing. So we're really excited that the RNA sequencing is done with a FMR1 genetic background. And yeah, so more to tell you later.
Thank you for the information.
Yeah, you're welcome. Thanks for coming, Frank.
Yeah.
Thanks, Frank. The next question comes from Miri Van Hoven. Let's get Miri's mic opened up. You know, she's going to ask a tough question. Go ahead, Miri.
Hi, Wendy. Great talk.
Hi, Miri.
Of course, you touched on my absolute favorite subject, synapses. And so I was so happy to see so many of my favorite genes in your lineup. And I'm excited to know what kind of follow-up experiments you and your collaborators are going to do with these lovely and exciting genes.
Yeah, so this is, I mean, everything is just happening back to back so fast. Right now, we're just focusing on understanding the gene expression differences between the FMR1 background with or without BPA. And then we'll then also compare between the wild type and the FMR1 background. And so once we identify, you know, genes that are absolutely relevant, then we might actually do some more, you know, studies, maybe knockout, or, you know, we haven't really talked explicitly about what we're going to do, but I can only imagine that we can totally go down and just work on a handful of genes and see what kind of impact it has on the neurodevelopment.
Oh, so exciting. Yeah.
Thank you.
All right, the next one is a technical question from Harrison Yu. Harrison, you can open up your mic.
Hi, so I would just like to know what kind of software is being used to analyze the sequences, because I saw the slide about using the department's high performance computing machines. So I was just wondering what kind of software you guys use.
Yes, so all the software that we use are open source, you know, publicly available bioinformatics tools. So for the sequencing reads coming out of the sequencer, we have done a bunch of pre-processing, including quality control using FastQC, trimming the adapter using the sequencing adapter that might get into the sequencing read using Trimmomatic or FastP, and then aligning the sequencing read to the transcriptome. We have to use a splice-aware transcript mapper such as STARS. I think Eden is using HiSet, but you know, I don't limit student using different tools as long as we get, you know, good results, because there are so many tools out there. And then for the differential gene expression analysis, we're using DESeq2, which is our R package. And gene set enrichment, we're also using an R package called FGSEA. And so it hits different databases just to see, you know, pathways, what genes are in which pathways, which gene set is in which pathway. So that's pretty much sum up all the tools that we use.
Thank you.
You're welcome.
Okay, it looks like there's a certain person named S. Curry who wants to ask a question. Let's see if I can open up this person's mic. Go ahead, S. Curry.
Hi, Michael. Hi, Wendy.
Hi,
Wendy.
Sammy. Thank you for coming.
Yeah, great talk as usual.
Thank you.
Yeah. Whoa.
It's my dog agreeing that it's exciting to hear your voice.
Okay. Yeah. So, as a follow-up to Michael's question, have similar experiments been done with other model organisms, not necessarily human? And if yes, did you get the same pathways, the same genes that are up-regulated, down-regulated as Drosophila melanogaster?
Yeah, so as far as I know that there are definitely other model organisms being used to study BPA exposure. And I have to say that I have to look more closely into if there are similar genes and pathways that are being identified in other model organisms. And so that is something, that's a great question. And I think that would be really encouraging if we see similar results in other model organisms, because there's a stronger support that, you know, genes that are orthodox doing the same thing, if it's impacted by BPA, have the similar impact. So, but yeah, that's a great question. I definitely look into that.
And I think your current chair, Melody, had a question as well. Melody, do you want to ask your own?
Sure. Thank you very much, Michael. And Wendy, thank you so much for your excellent talk. And I just want to let everybody know that Wendy right now is developing our CS22A, which is originally Python Introduction to Python for non-majors. Now it is going to become an introduction to Python for data science, which will be part of our course in a new degree program that we are developing, Bachelor of Science in Data Science, BS in Data Science. And Wendy is heading on this task. And so Wendy, my question is, every semester we have a MS Bioinformatic welcome gathering, and we invite all the bioinformatic researchers in computer science and also in biology and other departments to talk about their research. And Wendy usually has a slide, but with like 6, 7 projects, exciting projects that I knew nothing, but students are very interested in. So I'm just wondering if there is time Wendy can tell us lightly about these projects so that everyone here who's interested to know more about that or to collaborate can also get in touch with you.
Sure, absolutely. Well, thank you. That's a great question, because I definitely can get help from more students, because I am very greedy and I really excited about many projects. So let me tell you a few projects that I have. So one of them is actually related to this project. It's also from the same collaborator from Sacramento State. So I didn't show you explicitly this one gene, KISS. You see KIS right here. So in this lab, they have another study looking at a KISS null mutation, meaning the KISS gene is not expressed. And they notice that when this gene is not expressed, the flies is actually having learning disability and is having some similar to, like, severe ASD, autism spectrum disorder. So they want to study another environmental factor, which is gut bacteria, how that influence the brain when you have KISS mutation, like a null mutation. So that is an ongoing project. It's a metagenomics data analysis involving analyzing the gut microbials. And also they notice that they actually use antibiotics, wipe out all the gut bacteria and see if there's no gut bacteria, how does that influence the behavior of the fly as well with the KISS mutation background. So, and because we have the metagenomics data, we see a bunch of different organisms in the guts and different parts of the guts as well. And they also team up with neurophysicists looking at the movement of the gut tract, how fast the food goes down. And they decided to do RNA sequencing of the guts as well. So the different parts of the guts, they send the RNA out for sequencing. So we just got the RNA sequencing results back. So there are a lot of things going on. So just for flies in neurodevelopment. And then I have another project that also is ongoing. I am always really interested in knowing, we're using all these sequencing technologies, and how much can we trust that, right? I mean, it's a machine, there's a black, sort of like, it's not exactly a black box, but it's a box that spits out a bunch of data. How much can we trust all these data? Is there any mistakes? Can it be upstream? Sorry, let me go back to, where's my slide now? Oh, hold on. Is there any like upstream manipulation of the genetic materials, such as RNA extraction, isolating mRNA using, some molecular kit, will that somehow modify the DNA or modify the RNA that leads to a readout that's different? So my research focused on looking at sequencing artifacts and see if there's any treatment upstream of sequencing can also lead to mutation that is not biologically relevant. So that's another project. And yeah, so I have a few more projects, range from sequencing goats from Pakistan to pica from the Nevada Sierra with our home, biology department. Yeah, there are a few projects actually brewing. And so if you're interested, come talk to me. I am happy to, have further discussion and see, where you can fit in. Let me see. This is my email. I see Bill's hand went up.
Yeah, I'm sorry. Let's see, Bill. Oops, one more second. There we go. Go ahead, Bill. You can ask your question.
Hi, Wendy. Thanks
Hi,
for the
Bill.
talk.
Thanks for coming.
Yeah, I actually have an office hour in parallel. So. I'm curious, in the heat maps that you showed, in the columns, when you show the BPA exposed group, are those replicates or are they different?
They're biological. They're biological replicates. So they're not technical replicates. So they're different.
So they're different, like organisms, basically.
Right.
Okay. So do you think it would make sense to separate the organisms by change in behavior? Like, because you mentioned before that you also observed change in behavior after BPA exposure. So I don't know, some flies cannot fly maybe or something like that.
Yeah, so, we definitely, so I think, right, like for this particular RNA sequencing, we're just looking at flies that are exposed versus not exposed to BPA. But if we do further breakdown, like if we do observation, and then if the fly is still around, and it's actually, Phil, probably will be really hard because these are larvae. We're looking at neurodevelopment. So the only thing that we can see that is different, maybe we can segregate how severe the axon guidance disruption is, or, because these are not adult flies. These are actually the baby larvae. So there's not much behavior we're observing.
Okay, but you could still, okay.
Yeah.
Okay.
So because we're looking at neurodevelopment, so we want to get the larvae version of it.
Okay, and also, do you think it would make a difference if you vary the exposure, like if you?
Yes, absolutely. Yeah, because the BPA exposure is actually known to be not monotonic, meaning that it's not like the higher exposure, you're gonna get a more severe outcome. It's actually kind of like a U curve. It's interesting because I actually, at the end, I actually have a paper that actually show that non-monotonic dose response curve occur, and is really relevant because like the FDA using a very traditional way of toxicology, how the higher the doses is worse and that kind of thing, but not every chemical actually behave that way. So that's,
using GitHub.
Oh, sorry, I know you have office hour. Let's go back to GitHub then.
All right. I think that we have expended our questions, and it's a little bit after 5 o'clock, and I want to respect people's time. So Wendy, I want to thank you again for a great talk. I can see how being in bioinformatics would open up an infinite number of projects. Really cool and great for students, great for you. And I'm really looking forward to seeing future results. So thanks everyone for joining us. Thanks, Wendy, for a great talk, and hope to see you all again at another seminar.
Thank you very much.
All right. Take care, everyone.
Take care. Bye-bye.